I'm a pathologist. I'm going to show you lots of pictures, and I'm going to talk very fast, but if I have something important to say, I'll try to point that out. So let's just start with the belly of the beast. We've been talking about influenza A virus. I just want to show you an electron micrograph of real flu viruses up at the top, and then this diagram here. And I don't have time to talk about all the molecular complexity of the virus, but just a couple of important points. Flu viruses, the flu A viruses we're talking about, are RNA viruses. They encode their genes with RNA, not just as the messenger that you would use RNA for, but their genes are encoded. There's no DNA form of the virus, and so the virus has to replicate RNA to RNA. And unlike when you are typing on your cell phone or your Microsoft, there's an autocorrect or spell check. The virus lacks that ability to spell check, and it has a very high error rate. And it makes tons of mutations. So every time the virus replicates, it makes several mutations. You think this is a really bad lifestyle choice, but the virus has adopted this choice so that it has kind of every conceivable mutation possible all the time. And if the virus finds itself in a new species host, or it might have a mutation that would help it. If it finds itself under immune pressure from pre-existing immunity, it can escape from that. If it finds itself being treated with an antiviral drug, it can have a pre-existing mutation that allows it to escape from that. And as John said, the other important point is the segmented nature of the genome, so that new combinations of genes can continually emerge. Flu is a huge problem for people, but it's almost impossible to imagine controlling it because it's not just a human virus like measles or polio, smallpox, which have or could be eradicated. The problem with influenza A is that it's in probably hundreds of warm-blooded animal species, both birds and mammals. The largest natural reservoir are wild waterfowl, where it's a GI virus shed from their cloaca, as shown anatomically here. The thing about these viruses is that they can move around and cause either localized infections in new hosts that don't last, or actually permanently adapt to a new host species like horses or pigs or dogs or humans. And the mechanisms by which these viruses move around is incredibly complex, and we've done a very bad job understanding the molecular basis for what controls host species switches, but we know that it's a critically important aspect of what needs to be done. Flu viruses are the poster child for rapid evolution, and I juxtapose a sketch from Charles Darwin's notebook from the 1850s, where he says, "I think," and then he thinks about the evolutionary divergence through natural selection and speciation. And here is a family tree of the hemagglutinin gene of influenza A viruses, and you can see that Darwin, I think, would really like to look at the genetics of influenza. So they're incredibly divergent, incredibly rapidly evolving. So the important question is to understand ultimately how influenza viruses get from this orifice to that one. And we know it happens. It just doesn't happen very often. And it may happen through intermediate hosts, as John said. This picture, it's hard to know who's enjoying this interaction more. But it turns out that while pigs are classically considered a mixing vessel, it turns out that humans are much better at giving human viruses to pigs than pigs are to humans. And so in addition to being turned into bacon, pigs have a lot to worry about. There have been 14 pandemics that we know of in the last 500 years. The earliest really well-documented pandemic in 1510, but there have been probably others before that. In the last 100 years, there have been 4 that were mentioned, the biggest one, the 1918. And this is not a lot of different choices to understand how the complexities of different pandemics form, but there are some important things that we should notice about them. This we've already, Tom talked about that, the difference in mortality. I mean, if you take about the numbers, the population of the US was about 1/3 the size in 1918 that it was in 2009. So while there's about a 55-fold difference between mortality between 1918 and 2009 pandemics, you could think of that maybe as like a 150-fold change in mortality. So pandemics are incredibly variable in their impact. Here's one important lesson that I would like you to take away from. This is a very complicated slide, forget the details, but here's the story. An individual virus passed from a bird through some mechanism to adapt to people in 1918 or just before 1918 to cause a pandemic. That virus killed 50 to 100 million people in a few months. That virus never went away. that has continued to cause, and its descendants, to cause all human influenza in the last 100 years. So that all the subsequent pandemics and all the seasonal flu are all due to the single introduction of one successful virus into humans. So it's not just the tens of millions of people who died in 1918, but the millions and millions of people who've died in the last 100 years, all due to one successful introduction. So we are still in the 1918 pandemic era, 101 years later, and there's no evidence that this era is going to end soon. So these subsequent pandemics have just been genetic updates of the original 1918 virus. Since we're in Philadelphia, I mentioned that in the outbreak in Philadelphia, 16,000 people died, 11,000 just in the month of October. That's a pretty stunning thing to think about. A very poignant way to look at that are death records from the state of Oregon, bound one page for each death by month, and you can just see when the pandemic emerged in the fall of 1918 and when it goes away in the early months of 1919. So there were no virus isolates made in 1918, so there was no way to study the virus. So in the mid-1990s, at the Armed Forces Institute of Pathology, we had the crazy idea that maybe we could find tiny fragments of the RNA from formalin-fixed, paraffin-embedded autopsy tissue blocks from US soldiers that died, as well as from exhuming bodies from Breivik Mission, Alaska, where John mentioned 85% of the adults died in 5 days. So with Johan Holton, an exhumation was done in 1997 to obtain some tissues. The effort to sequence the genome of the virus using technology available to us then took 9 years. And then after that, a large multi-center collaboration involving many people, including Peter Palese, who we'll hear from later today, were successful in reconstructing the virus so that we could study it in the laboratory. I'm happy to say now, that because I'm old and impatient, that instead of doing 9 years, we can now sequence the genome of the 1918 virus in about a week from an autopsy tissue from 1918 using new technologies. So the ability to look at other extinct pathogens is much more practical than it used to be. So let's talk about the damage done by the virus. I'm going to show you some autopsy tissues of people who died in 1918. So these are hematoxylin and eosin stained sections of lung. The lung is mostly air. These very delicate, if you would imagine sectioning a sponge, these little air sacs are where oxygen exchange occurs, and then there are kind of the branches of a tree that move the air in and out. In 1918, in the people who died, there was really no airspace left. Their lungs were filled with fluid. They basically drowned, fluid or blood. There was a lot of damage to the cells lining the lung. Here you see this fluid at high level. And then there were lots of blood clots in the lung. While you can see that pathology that reflects the pathology of the primary viral damage, most people died with a secondary bacterial pneumonia. Probably 95% of people died with a secondary bacterial pneumonia, no antibiotics available. And so here you see the damage caused by the secondary bacterial pneumonia, just seas of inflammatory cells. And you can do gram stains on these tissues and just see the abundant bacteria, pneumococcus and group A strep, staph, many other species. You can also stain for where the virus was in the lung of 1918 victims, and the answer was that it was everywhere. It was from the epithelium in your nose all the way down to the smallest air sacs in the lung. The whole lining of the lung is stained brown here, which is the presence of the virus. We have studied about 70 1918 autopsies, including a number of them, about 9 or 10 cases, from before the pandemic was recognized in the fall. And now I'm going to briefly turn to the sort of perfect storm that the virus created to cause the kind of disease and mortality that we've been talking about. So there are viral factors, bacterial factors, and host inflammatory factors. So we start with the virus. This is the most complicated slide, even though there's no picture. I have to summarize a huge amount of work in a very quick time. So let me just say a couple important points. The 1918 virus is incredibly bird flu-like at a genetic level. We don't know how it went from a bird reservoir to humans, whether it went through an intermediate host, when or where it emerged, and we probably will never learn that. But it is very bird-like. It turns out that the hemagglutinin gene, the gene that encodes the major surface protein of the virus, the H1, is the major virulence factor. And there's no mutation in it that we know of that accounts for this incredible virulence. More scarily, it looks like it's just something that it's simply inherited from a wild bird H1 ancestor. So that currently circulating bird viruses share the same virulence properties if they adapt to mammals. And I'll show you that in a minute. So I can say that it is possible under very controlled circumstances in high containment labs wearing these fashionable suits to work with the 1918 virus safely or other high path viruses like bird flu. And here are a series of experiments in which we show that you can take a wild bird influenza virus, an H1 virus, from a contemporary bird and make a single nucleotide change out of 13,000 that makes the virus behave just exactly like the 1918 virus in terms of its virulent properties in mice and ferrets, in macaques and other animals. So what about other subtypes of the virus, since there are 16 subtypes? So all of them can grow in the lungs of animals, in this case mice, but cause mostly no disease, where a subset of these 16, this is weight loss, when infected, the mice rapidly lose weight and die in about a week. And they're subtypes 1, 6, 7, 10, and 15. And Stacy will talk more about this later. The thing that's scary about that is that viruses of those subtypes have caused human infections in the last decade, H10, H6, H7, and H7 is one that we particularly worry about. So let's talk about host inflammatory factors. This is another very important point. The virus itself causes damage, but a lot of the damage that you see in a lot of the morbidity and mortality is due to the host inflammatory response to the virus. Here you can see a typical seasonal infection causes no inflammation in the lungs, where here in 1918 you get this massive inflammation of white blood cells into the lung, leading massive upregulation of genes associated with the inflammatory response. And in a critical set of experiments, John Cashman's lab infected mice with a lethal dose of the 1918 virus, waited for them to get sick, and then treated them with a drug that has no antiviral properties at all, but it blocks a particular part of the inflammatory response. And those mice lived. They had no decrease in the amount of virus in their lungs. It's just that they did not have the inflammatory response. They had less free radical oxygen damage and survived. So it shows you that the host response itself is a critical component of the disease in 1918. So let's talk briefly about bacterial factors. As I said, it's probably impossible at this stage to know exactly when and where the 1918 virus emerged for human spread, but I would think probably somewhere under the season of the flu season before 1918, that is the flu season of 1917 to 1918. And by the summer of 1918, the virus was clearly in North America, Europe, and Asia. In a study of US military camps, there was a teeny tiny, just an unbelievably tiny number of fatalities in the summer months before the pandemic was recognized. This is a log scale, so the number of deaths here is incredibly tiny. But if you look at those very few cases, they have the autopsy findings exactly what we were talking about, influenza with secondary bacterial pneumonia. So this particular kind of disease progression, I think, is a really sensitive epidemiologic biomarker of a new pandemic emerging, before it was epidemiologically visible. So what happens when you have a secondary bacterial infection? The virus goes through and clears off the cells that line the respiratory tree, exposing these stem cells, these basal stem cells, which rapidly reproliferate and reproduce the epithelium of the respiratory tree. They expose binding sites for bacteria, and in the absence of the bacteria, these cells rapidly reproliferate and repair the lung. When you add bacteria at the right time, you kill off these cells, and the lung can no longer repair itself. And what you get is a loss of genes associated with repair and reproliferation. In the case of 1918, it was really a particular perfect storm, in that the 1918 virus itself, very unusually for a virus, induces a huge infiltrate of a kind of white blood cell called neutrophils that are very damaging. In the case of bacterial infection superimposed onto 1918, you get this unbelievable amount of inflammation in the lungs. And this actually makes the bacteria more virulent because they're responding to this inflammation in a way that kind of upsets them, and they're trying to defend themselves, and it ends up being more virulent. So you have this kind of perfect storm where you actually have upregulation of genes in bacteria in response to the inflammation from the virus. And this leads to kind of the fulfillment of Virchow's triad. You activate neutrophils, you cause damage to not only the epithelium, but the vessels of the lung, and you get widespread clotting. And you see this not just in animals, but in human autopsy tissues. And you wonder if the kind of heliotrope cyanosis, the bluing that John Barry referred to at the beginning, relates to the massive amount of blood clots that occur in the lung following a 1918 infection. Curiously, just as an aside, one of the cases of 1918 that had blood clots we looked at had very interesting features where the red cells were sickled. So we went back and looked at the records. This was an African-American soldier who died in October 1918. And we went back and sequenced the beta-globin gene from his DNA and showed the classic sickle cell mutation 4 years before the term sickle cell anemia was coined in a paper published in 1922. So in the last few minutes, I want to talk about what do we do to prepare for a new pandemic in the future. We don't yet know how to predict when a pandemic will occur, how virulent it will be, what subtype it will be. We can only respond to a pandemic once it has occurred. So clearly we need to do a better job at understanding how genetic changes in the virus relate to its ability to move around between animal hosts. How does a virus go from birds to humans? I think a difficult observation is that it's not just a simple answer, that different viruses can adapt different ways. It's a multifactorial process, and so even if you understand how the 1918 virus adapted from birds to humans, it wouldn't necessarily give you the answer of how a new pandemic in the future would do so. But we need a better understanding of structure-function relationships. Clearly, we need to do a better job at vaccine development. 75% of influenza mortality in the last 60 years is due to seasonal flu, not due to pandemic flu. Everyone is so concerned about a new pandemic, and yet year after year, so many people die of seasonal flu. So impacting seasonal flu morbidity and mortality would be incredibly important. So while I think that studying influenza in the laboratory and in animal models is critically important to understand basic pathogenesis, I think that the most important thing as a physician is that we study influenza in people and to use that information to help us understand how humans respond to flu, the inflammatory response, the immune responses, and what we could do to use that information to make better vaccines. So I'm going to very briefly mention that we've been studying human influenza in my group at the NIH for the last 10 or 12 years. But in the last few years, we've been doing a thing called a challenge study, in which we take very healthy, carefully screened volunteers and put them in an isolation ward at the hospital at the NIH Clinical Center, and then infect them intentionally with circulating influenza viruses. And this allows us to look at self-limited influenza in healthy young people, but it is a really important way to look at immune responses, but it is also critical to help us evaluate new drugs and vaccines very efficiently in a cost-effective way. So we've done close to 500 patients now safely and effectively. We're looking at multiple viruses, A viruses and B viruses, and you can use that to evaluate new vaccines or other therapies. One of the things that we've learned, for example, is that the focus of influenza vaccines in seasonal and pandemic flu for the last 60 or so years has been the hemagglutinin protein, and that antibodies against that protein, either the head or the stalk, are important correlates to protection. And yet, sort of the unloved stepchild of influenza virus is the other surface protein, neuraminidase, and yet antibodies against the neuraminidase are actually a better predictor of protection from disease than hemagglutinin, and yet there's very little characterization of the neuraminidase in vaccines or to antibody responses against that in the last few years. So I think focusing on neuraminidase is another extremely important thing to think about. Most people who get influenza have self-limited disease and may feel terrible. You may be sick for 10 days, but you're going to get better. Clearly, there is a subset of people, both in pandemic and seasonal flu, who are going to develop pneumonia and have a very high morbidity and a higher chance of mortality. But if you see a patient in a clinic or the emergency room presenting with influenza, it's very hard to know who is going to have a self-limited course and who's going to go on to develop very severe pneumonia that needs intensive care and hospitalization. So one of the things we've been doing in the challenge study is to, before you have any symptoms of flu, but when you're still shedding a lot of flu in your nose in the first day, to take RNA from white blood cells, from peripheral blood, and make predictions of who's going to have the most serious illness. And we have a set of genes that very rapidly helps us differentiate who's going to have serious illness before they're even symptomatic. So we think that this eventually could become a point-of-care diagnostic test to help screen for who is going to need more support. In the last few seconds, Peter will be talking a lot about universal flu, but I just mentioned some candidates they're working on. In my lab, we have a vaccine candidate that elicits a broad kind of immune response against hemagglutinin, neuraminidase, T-cell responses, and is 100% protective against every strain of flu that we have evaluated in mice and in ferrets. Here's a challenge in ferrets against an avian virus that the vaccine does not have an H10 or an N7 in it. You completely eliminate virus in the lung. You completely eliminate this kind of 1918-like pneumonia with unvaccinated animals too, having completely normal lungs in vaccinated animals. So we're going to start clinical studies of this in the next couple months, about February, March 2020. And then we can use the information from the challenge study to iteratively evaluate new vaccine candidates. So ours, Peter's, anyone's. You could imagine that as new vaccines come on board, you can do studies in these challenge studies efficiently, look at the correlates of immunity, every kind of test you can think of, see how well it protects, see how well the breadth of protection is from different strains of influenza, and then use that information iteratively to inform design to help make new vaccines. We'd imagine that this would be a slow, iterative process, and that this would be critical for how we would move forward. So with that, I will stop and thank the people in my lab who've done this, and the support from NIH, DARPA, BARDA, and especially the Bill and Melinda Gates Foundation. Thank you.
Great. Okay, questions for Jeff?
Great talk. I'm Mark Moffat, Smithsonian and Harvard. I'm just curious, I'm a biologist, but I'm still kind of naive about this, but these viruses don't die of old age. So you talk about currently circulating viruses, but there must be some that are potentially still out there from 10,000 years ago somewhere, right? So.
Well, I think the thing to think about is that influenza A viruses as a lineage of virus have probably been around for, who knows, thousands of years possibly. They're clearly their major reservoir are birds, where they've been diversifying, and we don't understand all the evolutionary pressures on those viruses to diversify the way that they have. But they can sort of accidentally infect mammalian hosts. But they don't seem to be as well adapted to mammals. So we know that flu is in humans all the time. Flu can be in horses. But all of those came from one lineage. Early on, I showed you a tree next to the Darwin picture, the diversification of the hemagglutinin gene. You can put all those genes on one family tree because as different as they are, they all came from a common ancestor at some point in the distant past. So it's not that viruses go extinct, it's just they evolve so rapidly that you have this kind of continual evolution of flu viruses, and you have multiple strains co-circulating with different evolutionary pressures. So that's what I was trying to say about 1918. The 1918 virus is still here. The flu virus, if you get the flu this year, you're still getting infected with a kind of an updated version of the 1918 virus after 100 years of evolution in people.
Hi, I'm Holly Smith from Ann Arbor. A comment, my father was one of those physicians in 1918 who met people in the emergency room. In case you're confused, I'm not really 100 years old. I was born much later. He left a couple of remarks. He said that the influenza patient was terrifying, a word I never associate with him, that I never heard him say in my life, that they were, and this was Detroit, that they were often blue when they were brought into the emergency room, and that even the ones that survived ended up with empyema, a great pleural space infection that in those days would have been managed with draining chest tubes, and so they were in for a great, long, terrible time.
Indeed.
So that's it.
Thank you for sharing.
Linda Greenhouse, New Haven. So last fall, I was part of a study that was maybe the other side of the coin of your challenge study. I was recruited while I was standing in line to get my flu shot for a study of how the vaccine is taken up by older people. Mm-hmm. And I had to go give blood every couple of weeks for months as they were testing this, and I never got any follow-up or, sort of. I mean, I never got the flu, but I just wonder whether you know anything about that or what.
Well, there are a huge number of studies going on. I don't know about that particular one. I guess I should have said at the very beginning that I'm from the government and I'm here to help you. But it is an extremely important question. And so the problem that we face in designing better vaccines for seasonal flu or to prevent or mitigate pandemics is trying to understand why it is that you could be infected, say, with a measles virus as a child and have lifelong really good protective immunity, or be vaccinated as a child and have near 100% efficacy. Where for flu virus, a couple of mutations every single year, you're eligible to be reinfected year after year. Why do you not develop better protective immunity? What are we missing? It's not the virus. I mean, the virus is taking advantage of a loophole in our memory immune responses. And what is that? And we need to understand that. And I think it's not just flu. I think it relates to the fact that flu viruses only replicate in the outer cells that line your lung. In a sense, you can think of it, those are still cells outside your body, like your skin cells, or they're GI viruses that just infect the lining cells of your intestines. Those are the viruses that don't replicate inside your body, that is systemically, for which we have a really hard time making vaccines. And I think the lesson is that we lack somehow the ability of recall memory responses in these mucosal surfaces. And I think that's where the key is. I don't know that we can solve the problem immediately, but at least we know what to focus on. We need to induce better immunity. And then how does that change in age? As you live, you're exposed to multiple viruses which change every single year. And then there are different subtypes that circulate at different times in your life. So you develop a repertoire of immunity that's different depending on how old you are. So young people have different immunities. And so how do we make vaccines that would tailor that? And so you might think that you might need to have different vaccines for different age cohorts and different responses. So it's a very long-winded answer to say I have no idea.
So 10 years ago this month, Steve Morse and Paula Olszewski and I published a one-page article in Science magazine, The Next Flu Pandemic, What to Do Until the Vaccine Arrives. And so I welcome all of this development, the challenge program, and so on. But we still have the possibility of flu pandemics. And if we understood how the disease is transmitted, which we didn't 10 years ago, which we certainly didn't in 1918, it could be very helpful. And even a tiny fraction of the money that's going for vaccine research or West Nile, it would help, because all we need to do is to delay a couple of weeks until selective vaccination can be done to reduce the replication factor below R0 equal 1.5 or so, which is the maximum, to less than 1, which squelches the disease in its tracks. So what do we know about the mechanism? Is it transmitted by fomites, picking up with your hands? Hand washing would help. Mask would help with big droplets, or aerosols. Thank you.
Yeah, thank you. No, excellent questions and comments. Influenza transmission in humans is probably all of those things, that it's a small particle, large particle aerosol, fomites. I think people are infected probably in multiple ways. It's not just aerosol. So clearly, I mean, when the CD says wash your hands, that probably does impact your development. I think one of the problems in controlling influenza, and one of the reasons influenza pandemics or epidemics are so hard to control, is that you shed virus in your nose for a couple of days, maybe 3 days, before you have any symptoms at all. So you might be shedding hundreds of thousands or millions of viral particles in your coughs and sneezes, before you actually have coughs and sneezes, just in your breathing and exhalation and secretions to spread to other people. So you're not feeling ill. So you go to work, you're on the bus, you're on a train, and you're exposing other people. And this is one of the problems, that the first part of the spread of the virus is silent. And so it's very difficult to control using standard public health measures until the epidemic curve has gotten so high that everyone has it. So it's trying to control it down here in the curve is the real challenge.
Okay, well, let's thank Jeff once again. It was terrific, Jeff.